This is a short topic. We'll talk about the diagnosis and prevention of bacterial infection. And for at least some of you guys will be doctors in future for your career after graduation. And a proper diagnosis or accurate diagnosis is the basis for proper treatment. So if you have a patient coming into your clinic and you need to decide whether it's a bacterial infection or not. If it is, you want to decide what type of bacterial infection so that you can determine what type of treatment you need to give to your patient. So the diagnosis of bacterial infection is the basis. And also, we can also use like either medical or other procedures to prevent bacterial infection. That's some procedure you can use before the reaction can really happen. For example, immunization, like vaccination. Okay, first we'll talk about diagnosis. The bacterial infection diagnosis. Usually, with the development of microbiology, actually now there is a standard procedure or standard methods, a series of methods you can follow so that you can derive useful information for accurate diagnosis. The first is that, of course, you need a specimen from your patient. So you need to obtain a specimen from the infected site. You want to collect some tissue or some fluid from the site and then send the specimen to laboratory for analysis. And once the specimen is sent to the lab, and people working there will first identify the morphology of the specimen, whether they check it under microscope, whether there is bacteria or not. And they can start it whether the bacteria is moving or not. But as we mentioned previously that most bacteria are small, it's not easy to see their structure under light microscope. So usually they need to stain the specimen so that they can easily identify the bacteria. And because we can culture bacteria, we can culture some of the bacteria, we can culture to isolate the pathogen and then further identify or further determine the properties of that pathogen. So you make a pure culture and isolate the pathogen and then further analyze it. And we learn the antibodies and we learn the antigen-antibody reaction. And antibodies can recognize antigens specifically. So we can use this specific reaction and use the known antibody. You can produce known antibody and react your antigen to that antibody. If it can react, you will detect some antigen in the specimen. So this is a detection of antigen from the specimen by like immunologic assay, like ELISA. And detection and amplification of organism nucleic acid, that's DNA, in a patient's specimen. So that's with the development of molecular biology. We can detect the DNA directly, or we can also sequence the DNA. And also we can detect antibody in the patient's serum. If some infection happened, your immune system will respond to infection. and there might be some antibody produced and circulating in the serum. So we can analyze the serum and determine whether there is some antibody against some known antigen. So that's another way. Okay, first we collect the specimens. So, and of course we need enough specimens. So the quantity of material must be adequate. So you want to collect enough specimen for laboratory analysis. And second, the sample should be representative of the infectious process. Okay, so after the infection is onset, you want to collect the specimen. So, but you don't want to collect the specimen too late. If the patient has recovered, and if you collect something from the infected site, probably you won't detect any agent, any pathogen there. So you want the sample to be representative of the infection process. And you should avoid any possible contamination. And you want only analyze something in the specimen, but you don't want to bring in some other bacteria during the process, right? So that is a contamination. So you want to use sterile equipment and also aseptic precautions during the procedure while you take the specimen. So you don't want contamination. And next, the specimen must be taken to the laboratory and examined as fast as possible. So after you collect the specimen, you don't want to lay your specimen, leave the specimen in your clinic for a long time, and after a week you submit the specimen for analysis. That's not okay. So you have to take the fresh specimen to the lab and examine it immediately. And to keep the specimens fresh, we usually keep them on ice. And that low temperature can inhibit bacterial growth and can keep the specimens fresh. But that's for most of the specimens. Some specimens or some bacteria, they actually don't like the low temperature, because if you put them on ice, they will be killed, so that you cannot detect the organism anymore. So that specimens you don't want to keep them on ice. The meaningful specimens for diagnosis of bacterial infection must be collected before usage of antibiotics. Because if you have treated your patient with antibiotics and after a couple of days you collect a specimen, that doesn't help, because you used antibiotics previously, and that might kill all the bacteria, or you inhibit the growth of the bacteria, and then you probably will have nothing in the specimen. That won't help you to decide what type of agent is there. So this is a requirement of specimens, and you need to keep this in mind, because when you are a doctor to practice your clinical work, you probably will have to collect specimens and send the specimen to some laboratory for analysis, and you have to follow these procedures. And once the laboratory received your specimen, they need to analyze it. And the fast and quickest way to analyze the specimen is to examine it with a microscope. We can see the bacteria under microscope. And there are different ways. You can stain the specimen and give them color so that you can see it clearly. Or you can also check the live bacteria and see whether they can move or not, whether they can swim. If they can move, that means they probably have flagella, which can help them to move. So that's the bacterial motility you want to check. This is the method this is a method called Haring drop method. It's a drop of bacterial suspension placed between the cover slip and glass slide, and then you invert the glass slide and put it under microscope. So it's a drop hanging there, and that you can see whether the bacteria can swim or not, can move or not. I'd like to show you a video here. It shows us how to do the Haring drop assay, how to do the Haring drop method. There is no audio in this video, so it shows us the procedure. I'll try to explain the video for you. Let's see the video. Let's watch the video. So Haring drop method. This is you need to prepare the equipment and materials you need. And you have glass slides, and you need to, this glass slides. And you need to prepare some cover slips, the smaller piece of glasses. Next, you need to put some bacteria, a loop of bacteria, on the cover slip. So it's a drop there. You put a drop of bacteria suspension on the cover slip, like this. Then you need to sterilize the loop. Then you want to prepare some paraffin, and that's some grease on the glass slides. You put the paraffin on the glass slides, several drops, and this grease will be between the cover slip and the slide when you put them together, because it's grease. It keeps some space between the cover slip and the slide. See? You touch them together, stick them together, and then now you can imagine that drop is hanging on the cover slip between the cover slip and the slide. And then you take this cover slip and the slide under the microscope so that you can examine the bacteria in the drop. This is really an old type of microscope. There is no light source like a bulb. They use there is just a mirror to reflect light from outside, and you want to find the edge of the drop, because at that place there is some enrichment of bacteria. You can see here, you find the edge of the drop, and you switch the lens for higher magnification so that you can see the bacteria. And you can see the edge of the drop, and under a microscope you probably will see some particles there, and you can see whether the particles are moving or not. So that's the bacterial motility. And like this, you can see some bacteria and they are swimming happily. So with this Haring drop method, you can determine whether the pathogen, bacterial pathogen, is motile or not. Okay, this is the first method. And also we can stain the bacteria and check their morphology under microscope. And there are different types of stains, like simple stain, like the methylene blue stain, you can give the bacteria with a blue color. And also you have the gram stain, so that you can differentiate the gram-positive and gram-negative bacteria. And also there is acid-fast stain. And with this acid-fast staining, you can identify the mycobacteria. So that's the pathogen causing TB. Okay. The different types of staining. I think in your textbook, if you read your textbook in detail, you will find more staining methods. And this is the gram staining. We actually introduced this procedure previously. So these are the bacteria, either gram positive or gram negative. You fix them on a glass slide, and then you first stain them with a crystal violet, and then you stain it with iodine chemical, which can make the color more stable, and then you decolorize the bacteria with ethanol. And when you decolorize the bacteria, the gram-positive bacteria can maintain the color, while gram-negative bacteria will be washed pale. So there is no blue color left. And then you counter-stain the bacteria with safranin. And this safranin will show red color or pink color. But on the background of this blue color, you cannot see the red color for the gram-positive bacteria. And for the gram-negative bacteria, now there is no color, and when you counter-stain the bacteria, they will show pink or red color, so that finally you can tell the difference between gram-positive and gram-negative bacteria. So this is a very classical staining method. Okay, so this is the first step. You examine the bacterial morphology under the microscope. And we can also further isolate and identify the bacteria by culturing them. You know, when we culture bacteria, we need the bacterial culture media. And there are various types of media, including broth media and solid media, and containing different components. And each is designed for some purpose. And you need to pick the proper media to culture or isolate the specific pathogen in the specimen. So you can use blood agar, which contains 5% of sheep blood. And we can also use chocolate agar. Well, its name is chocolate agar, but it doesn't contain any chocolate. But it shows chocolate color. The chocolate color is due to the containing of heated blood. It contains hemoglobin, and those shows the chocolate color. And we can also use some selective medium, which can enrich the growth of some bacteria. And we can also use anaerobic media, and that's a media designed for anaerobic pathogens, which are growing in conditions without oxygen. And after you culture the bacteria, you can further examine the colony morphology, like their sizes and their shape, their texture, their color. And based on this, you can derive useful information for diagnosis. Whether the colony size is big or small, is it smooth, is it rough, is it round, or is it like a fried egg, and what color is it? Is it red or like pale white? So all these informations can be used for diagnosis. So this is the isolation and further identification of bacteria. And of course, when you purify the pathogen, purify the bacteria, you can further identify the bacteria by other characteristics, like cultural characteristics, like whether they require a unique nutrient, or whether they produce some special pigment, or you can use their biochemical characteristics, like the ability to decompose a special substrate, some sugar or some other substrate, or whether they can produce a particular product, like whether they can produce acid, whether they can produce gas when you culture them. And also you can identify their antigens with antibodies. And if they can produce a toxin, you can examine whether there is toxins produced. You can use models, animal models, put that toxin to the animal, whether you can check whether the animal is getting sick or not, or you can use cell culture to examine the existence of toxin. And you can check whether they are sensitive to antimicrobial agents. This last point is actually very important, because if you find some bacterial infection, you want to know whether they are sensitive or resistant to the antibiotics in your clinic. If they are resistant, you cannot use that antibiotic. Only if they are susceptible or they are sensitive to some antibiotic, you can use that one. So this is also important. It's a pity that you are not on campus. If you are here on campus, you would carry out several experiments, and that will include the test of antimicrobial agents when you culture the bacteria. And also you will do the gram staining, and also you will make a culture medium, make the dishes and culture bacteria on the dishes. And for antigen detection, we mentioned that you can use ELISA or agglutination. And these two experiments, if you were here, you will do ELISA. And agglutination is another experiment we're not doing right now, but previously we have this experiment. So ELISA, the full name is enzyme-linked immunosorbent assays. There are different types of ELISA. The most used one is called sandwich ELISA, like this one, sandwich ELISA. So what people do is that people use a capture antibody which can bind to the surface of a plastic plate. It's captured there, and this antibody is fixed on the plate, and then you add in the sample. If the sample contains some target protein, this might be a bacterial protein, and this capture antibody will bind to the target protein. That means we capture the protein. Then you add in another antibody. This antibody is called detection antibody. Sometimes this antibody links to an enzyme, this HRP enzyme, directly, or you can use another HRP linked antibody to bind to the detection antibody to form this structure. You can see that there is a layer of antibody, a layer of target protein, a layer of another antibody. This is like the three layers of sandwich. So that's what we call sandwich ELISA. And after the formation of this sandwich structure, we add in substrate, the TMB substrate. And the enzyme can catalyze the substrate and form some product showing blue color. So that according to the blue color, according to the color change, you can determine whether there is target protein or not. Because without the target protein, there is no sandwich structure formed. You cannot see the color change. Okay, I'd like to show you a short video to learn about the ELISA method. And in this video, actually, it shows us different types of ELISA. but it will also talk about the sandwich ELISA. Yeah, you can use ELISA test for AIDS, for HIV. Okay.
We have covered direct and indirect ELISA. I hope you now understand the basic difference between these two types of ELISA. Today we will study most commonly used format of ELISA, which is sandwich ELISA. Sandwich ELISA is used for the detection of antigens. Most popular application of sandwich ELISA is point-of-care testing and home test kits such as home pregnancy test kits. Sandwich ELISA uses two different antibodies for the same target antigen. Important point is that these antibodies recognize and bind to different epitopes on the target antigen. Let's understand the details of sandwich ELISA. Sandwich ELISA requires three main components: capture antibody, target or test antigen, and an enzyme-linked antibody. Thus, in sandwich ELISA, an antigen is trapped between two antibodies. In the first step of sandwich ELISA, capture antibody is immobilized in the microtiter well. Capture antibody is a high-affinity antibody, specific to the target antigen. Its function is to capture the target antigen from the test or serum sample. So, after the immobilization of capture antibodies, the microtiter well is rinsed. This is done to remove unbound antibodies present in the well. In the second step, serum sample or test sample is added in the well. Now, serum from a patient may contain many different antigens, but the capture antibody is specific for a single target antigen. If the sample contains the target antigens, they will bind to the capture antibody present in the well. If the specific antigen is absent in the test sample, no antigen-antibody binding takes place. Again, the well is rinsed to remove unbound and non-specific antigens. In the third step, an enzyme-linked antibody is added in the well. The enzyme-linked antibody binds to the antigen. Recall that these antibodies bind to the epitopes, different from those bound by the capture antibody. After some time, well is rinsed to remove unbound enzyme-linked antibodies. In the fourth and final step, the substrate specific for the enzyme linked to the antibody is added. Color development confirms the presence of the antigen in the test sample. Sandwich ELISA can be further divided into two types: direct sandwich ELISA and indirect sandwich ELISA. This classification is based on whether the enzyme-linked antibody is directly or indirectly detecting the antigen. Here, the target antigen is being detected by the conjugated antibody itself. So, this is direct detection. But here, a primary antibody is first binding to the target antigen, and the conjugated secondary antibody is detecting the antigen-bound primary antibody. So, this is indirect detection. That's all in today's lecture. In the next video lecture, we will understand how pregnancy home test kits work. Thank you for watching.
Okay, that's ELISA. And if you are on campus, you will also do this experiment, and you will do ELISA to detect the HCG. So that's also for the pregnancy test strips. And you can also do this strips experiment in the lab if you are back, probably in future. Okay, so this is the detection of antigen from the specimen. Another method is called the latex agglutination. Latex agglutination test is also relies on the interactions between antibody and antigens. So this method is carried out like this. So you have a latex particle or latex beads. You coat the beads with antibody, so that this is the antibody, the Y-shaped antibody. You stick the antibody to the beads, because the antibody can recognize antigen. If you mix them together, the antibody will recognize the antigen, the red one, because on each antigen there are multiple sites recognized by antibodies. And finally, with the antigen in the middle at the bridge, you linked different beads. So you linked all the beads together. It will form a big complex, and this big complex will come out of the solution quickly, so that you can see the beads stick together, and this process we call the agglutination. So this latex agglutination test to detect antigen in the specimen. This is another assay we used a lot. And when you are a doctor, when you practice your clinical work, probably you will have a lot of agglutination test strips in your office, and that can help you to get results in minutes. And for molecular diagnosis, that's another level, and with the development of molecular biology, and there are different types of methods developed. We can amplify and detect the existence of DNA or RNA molecule in the specimen, such as we can identify bacteria using their 16S ribosomal RNA. We can use DNA hybridization, or we can use PCR, that's polymerase chain reaction. I guess you understand the meaning of this one. Or we can directly sequence the DNA, the DNA chips, which are still being used for research or for clinical work. So this is the level of DNA detection. And another level is on the other side of the infection procedure. So if the host is infected, the immune system will respond and produce antibodies. So this serological test is to detect whether there is antibody in the serum. So we take the blood and detect the presence of antibodies against a microorganism. And you can carry out this detection in different ways, including ELISA, agglutination, precipitation, complement fixation, or fluorescent antibodies. And I'd like to introduce a method that's a complement fixation. Complement fixation is carried out this way, that you have your patient's serum here in the tube. You know, it's possible that the serum contains antibody against antigen, or it does not contain any antibody. Then you mix the antigen, this known antigen, and complement molecule with the serum. Antigen, complement, and serum. Mix them together. If there is antibody against the antigen, the antibody will bind to the antigen and also fix the complement molecule. This complement molecule we learned on immunology classes. So it fixes the complement molecule in this complex. This is with antibody, but without antibody, the complement is still free there. So after this, we add in red blood cells. So this is a sheep red blood cell. We add in sheep red blood cells and also add in the antibody to sheep red blood cell. If there is no complement available, no free complement molecule available, the antibody can bind to the red blood cells, RBC, but there is no complement. So this red blood cell cannot be lysed. But in this tube, there is complement available. After you adding in these two components, they will form a complex. And in this complex, the complement will lyse the RBC so that the color of this tube will change to red or pink. And this is a negative test because there is no antibody against the antigen. And this is a positive test because there is antibody against the antigen. And this procedure we call it complement fixation test. Okay. I do have a video here, but we won't watch it on class. You can always visit our Superstar application and watch it on our website in a class. Another method is using the fluorescent labeled antibody, so that you can use this antibody to detect the pathogen, and then you can check whether there is fluorescence under either microscope or use other equipment, and that will help you to find the pathogen in the specimen. We have another video talking about isolation and identification of microbes. This is a long video, about an hour, so we won't play the video here, but you can find this video in our class and watch it online. Okay, so this is the topic on diagnosis of bacterial infection. The second part of our lecture today is on the prophylaxis of bacterial infection. So prophylaxis means prevention. So what type of procedure we can take to prevent bacterial infection? And this is what we are talking about. You know, we learn immunology. We know that you can use vaccine to stimulate the immune system so that they can get some protective function and to prevent further bacterial infection. And immunization, we can use either active immunity, the active immunity, or we can use passive immunity. For active immunity, you give the host vaccine or toxoid. These are the antigens. These are the vaccine antigens, which can directly stimulate the immune system and let the immune system to recognize the characteristic of the vaccine or of the pathogen, so that when the pathogen really comes, and your immune system can clear them out or kill them quickly. Another one is passive immunity. This one, we give the person with plasma or globulin containing antibodies, and these antibodies can provide some protection or can help the body to fight against the infection. So this is passive immunity. Let's learn about active immunity first. For this one, we need vaccines. There are different types of vaccines, including the live attenuated vaccines. This, you are using the live bacteria, which are not virulent. They are live bacteria, but they don't cause disease. So that they can stimulate the immune system, but not causing real disease in the host. So this is the first one. Another one is inactivated vaccine. Inactivated means killed or dead. So you prepare the bacteria and you treat them with some physical or chemical method or heat or chemicals to kill them. And after you kill them, and they don't cause disease, but the components in the dead bacteria can stimulate the immune system. And you can use that as a vaccine. Or we can also use subunit vaccine. So you can use the subunit from the pathogen. Maybe it's a protein, maybe it's a sugar molecule, like a polysaccharide, and use that specific polysaccharide or protein, you can stimulate the immune system. And this is subunit vaccine. Now, with the technologies development, we have even more types of vaccines, like DNA vaccine, RNA vaccine. DNA vaccine, you give the patient, the host, some DNA molecule so that they can express and express some antigen so that they can stimulate the immune system. But for DNA vaccine, there is a risk that the DNA might insert into the host DNA, which can cause trouble. But you can also use mRNA, the messenger RNA, which is not long-lived, and mRNA cannot be inserted into our DNA. So it's more safe and easy to control. So the two vaccines approved by FDA in USA both of them are mRNA vaccines, and they work pretty well. And this is a real progress in this field. That and these are the first two mRNA vaccines available in history. And it is hopefully that in recent futures, the mRNA vaccine technology will be used in other fields, like to treat cancer and to treat infectious disease, and a lot of might be applied in different areas even for treatment in hospital. So this is the active immunization. And talk about the vaccines, we need to talk about the toxoid again. And we introduced toxoids before, and they are derived from exotoxins. But we treat the exotoxins with formaldehyde so that we can denature the protein. They are not toxic anymore, but they maintained their antigenic property. So we can still use that modified exotoxin and stimulate the immune system to induce some protective immunity. Passive immunization is that we artificially use a serum or antibody, either from immunized person or immunized animal, so that the antibodies in the serum can provide passive protection. And this is the passive immunization. We won't talk about that in detail. Okay, there is another video on prevention of bacterial spreading. We won't play it here because we ran out of time. And another topic is about herd immunity. Herd immunity, probably you heard about this word during the pandemic because in some countries they are using the herd immunity strategy to fight against the pandemic. That's a form of immunity that occurs when the vaccination of a significant portion of a population or herd provides a measure of protection for individuals who have not developed immunity. So this is a long sentence. It's not easy to understand. Let me show you a figure and to understand herd immunity. So herd means a group of people, a group of animal. Like this, you have a group of people here. It's a population. And if no one is immunized, it's shown in this blue color. And there are 2 persons carrying infectious disease into this population. And that agent can be spread from those patients to other person. There is person-to-person transmission. And because no one is protected, and finally a majority of the population get infected. So that's no immunization. But once there are some people immunized, they are protected. That's the yellow person showing yellow color. They are immunized. They are protected. But because the immunized person, the number is low, the portion is low, it cannot set a barrier to stop the spread of the germ. So finally, most of the people get infected. Only those immunized persons are protected. So this is another scenario. A different case is here that in this population, a majority of people get immunized, so they are protected. At this case, some of the person cannot be immunized. They are not immunized due to different reasons. For example, someone might be too old, their immune system is not working well, or someone is sick and the immune system is immunized, or someone is too young, it's a newborn baby, which cannot get immunization. But in this group, because a majority of the people get immunized, even someone carries the infectious agent into the population, and around this person there is a barrier of immunized individuals, they cannot get infected, so they cannot transmit the pathogen in the population, so that they can protect those non-immunized individuals. And finally, in this group, a lot of people get protected. Even those who are not immunized, they are safe. They are relatively safe. So this is how the herd immunity works. Okay? I think we can watch this short video on how the herd immunity works. So, well, generally it's the same thing like what we talked about according to the picture. Okay, let's watch the video.
In the absence of vaccination. First of all, let's observe the progression of a disease in a naive population, meaning a population that has never been exposed to a disease-causing pathogen or vaccine. The red zone represents the spread of the disease. Infected individuals are shown in red. The characteristics of the spread of every pathogen, virus, or bacteria are unique. Coverage of 50%. Now let's look at the same situation, but this time with 50% of the population having received a vaccine. This would be termed as 50% vaccination coverage. Vaccinated individuals are circled in green. The propagation of the disease is totally different. The individuals on the right have been protected. In the end, part of the population was protected directly by the vaccine as expected, and another part of the population was protected because it was not exposed to the disease. It is the surrounding individuals that blocked the spread of the pathogen. The phenomenon that provides this extra protection is called herd immunity. Here, the individuals protected by herd immunity appear in violet. Coverage of 80%. In the third hypothesis, the level of vaccination coverage is brought up to 80%. The spread of the disease is rapidly halted. Herd immunity is in full swing, and a large part of the population is protected either directly or indirectly. This is the reasoning for high vaccination coverage. Indeed, high vaccination coverage directly protects those who can be reached by the healthcare system and indirectly protects those who are less advantaged and cannot be reached. This is why vaccines play such an important role in public health policy.
Okay, so this is a herd immunity. When vaccines are available against coronavirus for the pandemic, according to the model, according to calculation, only if 75% of the whole population got immunized, we can end up the pandemic. We can stop the pandemic. So, if possible, if the vaccines are available in future, try to get the vaccine as soon as possible and try to get vaccine for your family, so that not only protect yourself, but it will protect the whole population in your community or all over the world. And everyone should receive a vaccine finally. Don't believe any saying that vaccination is not safe or not, because we are professionals, we are medical students and teachers. We should persuade anyone we know to receive the vaccine as soon as possible. Okay, so I think that's all for today. We covered a lot of stuff. This weekend we won't have a test, so you can take a break. I'll prepare the test, and it will be available for next weekend. Okay? All right. That's all for today. Have a nice day.